A bioinks is a formulation in which you have cross-linkable polymers which are biocompatible, and these materials can be combined with cells in a printing process, and with that you can actually replicate a scaffold design perfectly with your printing technique. Gelatin is actually a protein which is isolated from collagen, and collagen is the most abundant protein present in the human body. It has the advantage that it is cell interactive, and so is gelatin. So this means when we have a construct based on gelatin and you see cells on this, that cells will recognize it because you have a cell interactive sequence combining 3 amino acids, RGD, in there. And the additional advantage is that it's a very cost-effective material. It's actually isolated from slaughter waste. So it means it's actually also a sustainable product, and it's already around for many, many years in the pharma industry, as it's also used as capsules for encapsulation of drugs. And so basically the gelatines can be functionalized with cross-linkable groups, and these permit us to stabilize a 3D printed construct and also tune the mechanical properties of the construct that we form. And with that, we can actually also tune how fast a construct is degrading in the human body, so in vivo. Gelatin is already approved in different medical devices. The fact that it actually has a lot of different side chain functionalities means it's also offering different possibilities to functionalize it. And in the past, we actually already have shown that it's very compatible with cells. You can combine it with different cell types, and it supports cell growth, cell differentiation in case of stem cells. So it's actually a material which is cheap, readily available, and which we can tailor with chemical steps in order to ensure the best properties of the printed constructs.